Okay, so let's finish up now chapter 4. Chapter 4 deals with our nucleic acids. We know that we have two types of nucleic acids. We have DNA, the boss molecule, and we also have RNA. So this lecture, this discussion is only a few slides long, so it's not going to be a real long discussion. But we now address the idea of a second macromolecule, or I should say a second category macromolecule, a second nucleic acid that also has a role in genetics, but a different sort of responsibility, right? So if we use the analogy, like the DNA is the big boss, DNA is the president, right? RNA would be like the second in command, right? So the vice president in this situation. So it's a boss molecule, but it has a boss, right? So the vice presidents here, pretty high up, but they have bosses, the top level, right? And that's sort of the idea of DNA. So in this situation, we get not the gold medal, not first place, but the silver medal for second place. And we look at the idea now of RNA, ribonucleic acid. So what makes this ribonucleic acid? Well, there's a couple of sort of features that make this different than the DNA molecule. So first and important, RNA is going to consist of only one strand, only a single strand. So, and I hope you understand, single means one, right? So RNA, single strand, so that means it's going to be smaller. It's not a big double-stranded molecule. It's only a single helix that's sort of spiraling around. The fact that it's a single strand, the fact that it's smaller, lighter, faster, it's going to make it a better messenger. So a lot of times we're going to see this notated as RNA, but we'll see it oftentimes with a little M out in front. And that M stands for messenger. Right? So RNA, commonly referred to as messenger RNA, because that's what it does, right? RNA is going to take that message from DNA. Let's say, I don't know where you work. If you work, let's say that you have a meeting, right? And your boss is giving you instructions, giving you orders, but they didn't come from that boss. They came from the boss's boss, right? If you have little siblings, I bet you act as RNA sometimes, right? So mom tells, go tell the little brother, go tell this little sister to go get ready for bed. Go tell your little brother to clean his room. Go tell your little sister to, to wash to brush her teeth, whatever, yeah? So you are conveying that instruction to somebody else. And that's that role of the RNA, right? Now, if you were a big 800-pound beast, whatever, right? And they tell you, hurry, go tell your little brother to brush his teeth. Well, it's going to be difficult if you're very big and very slow to convey that message, right? So we want to be smaller. We don't want to be double-stranded. We want to be single-stranded. So size is going to be a factor here. Let's say you ordered a pizza, right? Would you want a big semi-truck to deliver your pizza? It could do the job. Or would you want a little car or even a delivery person on a motorcycle, right? So the smaller the vehicle, the quicker they can navigate through traffic, the quicker they're going to get you your pizza, right? So that's sort of the idea. So we want to take this messenger molecule, this RNA, make it smaller molecularly, lighter molecularly, so it can transmit the instruction a little bit faster, a little bit easier. It doesn't get clogged up in traffic. Another difference between the RNA molecule and the DNA molecule is the type of sugar. We have that nucleotide, phosphate connected to a sugar, connected to a base. Well, DNA has deoxyribose. RNA is going to have ribose sugar. So it's a different type of sugar. It's got that hydroxyl group on carbon 2. I don't know if you're fans of lucha libre, wrestling, right? WWE, that kind of stuff. So if you are part of a tag team and you're getting beat up, right? Oh, I'm getting beat up. Let me go tag out so somebody else fresh can come in. So if we can kind of use that analogy a little bit here. So if you remember the 4 bases in DNA, right? The DNA molecule, we have adenine, we have adenine binding with thymine, we have cytosine guanine binding with guanine. Well, in this situation, and I'm not going to get bogged down in the biochemistry here, but in this situation, we're going to say thymine tags out and uracil tags in. So RNA, also like DNA, has 4 bases, but they're a little bit different, right? The adenine and the guanine are the same. The purines, we have adenine and guanine in DNA, we have adenine and guanine in RNA. So the purines remain unchanged. The pyrimidines change. The pyrimidines in the DNA molecule are adenine and thymine. The pyrimidines in the RNA molecule are adenine now and uracil. So uracil now becomes a factor in the RNA molecule. And again, why do you think this is the case? Uracil is a little bit smaller. It's a little bit molecularly, it's lighter by a very little bit. So we're trying to make this thing smaller, faster, easier to navigate through traffic. So with all this, right, what are we trying to accomplish? We're trying to make it simple for the big boss. The big boss doesn't do work. Again, hey, RNA, come over here. RNA, you go out and you do this over there, right? Take this message, go make some proteins over there. We need to repair this injury over there, so go, take care of that. So the big DNA, the big CEO, the big Jabba the Hutt molecule doesn't do much. It commands RNA to go command somebody down the line over there. That's sort of the idea. So maybe not quite like normal, but I think it can be done now, right? If we were to go out to a nice restaurant, you could sort of see this progression, right? If we go to a nice restaurant, you would be analogous to that DNA. Right? All you do is you go and you sit down, right? Somebody arrives at your table. May I take your order, right? So the waiter, the waitress arrives at your table, and you can analyze the situation, right? So you're not going to cook the food. You're not going to do any work. You're going to go and tell the waiter, the waitress, I want this dish. I will take this entree. Yeah. Well, the waiter, waitress is not going to cook your food. They're going to take that instruction to to the chef in the back, right? The chef is the one that's actually doing the cooking of the meal, right? It's not to say, I know waiter and waitresses do a lot of work, right? Don't get me wrong, but they're not the ones actually cooking the meal, right? So the DNA person gives the order to the waiter, the waitress. The waiter, waitress takes the order and gives it to the chef. So the chef is the one that's actually cooking, and that's what we say. So RNA decodes DNA information to help make proteins, right? So the RNA doesn't make the proteins, it transfers the instruction to the appropriate, we're going to call this a ribosome, we'll get into that in a bit, to another part of the cell where that protein can be made. So that's sort of the role for RNA. And in this situation now, this is just a very short section. Remember, DNA is 1 million base pairs long, very big. We just have a very short section, this double helical structure, G with C, A with T, A with T, C with G. So we have this going on. Phosphate, we know that this would have to be the deoxyribose sugar, right? We have thymine, so we know we're talking about DNA. Now down here, it's not listed, but we know this sugar would have to be ribose, right? Ribose sugar, phosphate sugar. So it's the same repetitive type of backbone. Hey, there's a U, right? So if we're dealing with uracil, you know we're dealing with the RNA molecule. Right? Now, let's see if you're grasping this concept, right? So we know we have our complementary matches. What do you suspect would be matched up here? So we have a G, we have a U, an adenine, a guanine, a cytosine, an adenine, a uracil. So what would match up with this U? What would match up with this A, with this G, or with, I'm sorry, with the C, with this G, with this A, with this U, with this G? What would match up there? Did you get it right? Yeah, you sure? Did you tell me that something is going to match up here? What would go there, right? If you tell me nothing goes there, you got it right. This is a single strand, right? You just told me over here. Yes, yes, I know, single. Single means one, yes? And some of you try to tell me some sort of letter that's going to go here. No, nothing will go down here because this is a single strand of that molecule. Is finished, it's done. There's no complementary match with the RNA molecule. There is a complementary double strand for DNA, but not for RNA. Remember, we're trying to make it smaller, easier to fit through traffic, right? That's the whole point. Instead of being a million letters, it will be a short section, right? A short little section of molecule. So again, I hope that makes sense. That's the information that you're going to need to, to navigate through the exam, through the homeworks, that kind of stuff. So this next bit, I'm going to preview what comes next. So we're going to kind of hop. We're going to make a big hop or a big jump. Right? We go from chapter 4. We move way ahead to the idea of this dogmatic type of idea. So something, I believe it's chapter 12. And maybe I don't have the book right in front of me right now, but we're going to hop over to a chapter that deals with the actual formation of the proteins. Right? So I want to sort of elaborate on this term dogma. A dogma, it doesn't refer anything to canines, to pets, right? A dogma is basically, this is a definition I just Googled here, but a dogma is defined as a principle or set of principles laid down by an authority as incontrovertibly true. Okay, well, let's dissect this out, right? So a dogma, a principle, so some, ideology, some set of governing laws maybe, laid down by an authority. So some authority figure, some boss figure, right? As incontrovertibly true. What does that mean? Incontrovertibly true. So if something is incontrovertibly true, we cannot challenge that. You can't say, excuse me, I think that's no. You can't challenge, you can't argue. If you are going to believe, if you're going to be part of that dogmatic sort of ideology, you must accept everything, or if you don't accept this bit, I believe 90% of it, but 10% I don't. Well, then you don't really practice, you don't believe that whole dogmatic sort of entity there. Right? The best example I can give you is like different types of religions, right? So religions are quite dogmatic. Be it Christianity, Catholicism, Buddhism, whatever religion that you practice, if you practice religion. So in this situation, let's analyze Buddhism, right? So if somebody is a Buddhist, to be a Buddhist implies that you follow certain rules laid down by whoever invented that Buddhist religion. Yeah? So for the most part, Buddhists are very, very passive. Part of their ideology is that they have to be passive. So imagine you're a Buddhist, right? So I'm a Buddhist. I'm gonna practice everything Buddhists do. But if somebody hits me, I'm gonna turn around and punch them back. So, well, you're then not following. You're not following that passivity. You're not following the rules laid down by the dogma, right? If let's say if you're a Christian, if you're based in Christ, right? Well, I'm Christian, but I don't really believe this part, or I don't really accept this part of this teaching. Well, then you're not following that dogmatic sort of religion there, right? So the dogma itself serves as part of the primary basis of an ideology or belief system. The dogma cannot be changed or discarded without affecting the very system's paradigm or the ideology itself. Yeah. So let's say, right, Christianity is probably the most common religion here in the El Paso area, but let's say, well, I'm Christian, but I don't believe in I don't know, I don't believe that you should be able to break the commandments, right? So commandment 1, don't kill. Well, I believe that I'm a Christian, but I believe if somebody pisses me off, I have the right to kill him, right? Well, you're not following that dogmatic teaching, right? So the ideology itself is not being followed. So what we're trying to do now, I'm trying to establish this dogmatic idea that whoever established the religion made this important right here, incontrovertibly true, right? So one thing about religions, right, is something that you believe, some moral, some ethic ideology. We cannot challenge that religion, right? You can't go and talk to the preacher. Hey, preacher, I think you need to change this rule. Right? No, it's based on however it was laid down. When we talk about biology, we do have a process. I don't really like the phrase that they use here, but in science, right? Nothing in science is incontrovertibly true. That's one thing I like about science. So science is based upon data. And if the data shows that, no, you know what, this is not right. Well, we are able to change our ideology. If we go back to the DNA molecule, in 1952, proteins are the basis of genetic information. 1953, oops, we got that wrong. Not proteins, is actually DNA. So we rewrote the biology, not me, but whoever was around back then rewrote the biology textbooks to encompass new information. Right? That can't happen with dogmatic religions. So when we talk about the central dogma of biology, the central dogma is a dogmatic ideology, but the ideology is allowed to be changed depending on what new information arises, right? So right now, the framework of biology in the year 2020, and has been since 1953, is that DNA is that central core molecule, right? We inherit DNA. Once we inherit DNA then is going to be transcribed to form RNA, that single-stranded molecule, and that RNA is then going to go help us form proteins. So it's a progression of molecules. Two categories. We have nucleic acids and proteins. When mom and dad conceived us, right, they passed down not the proteins that we are today. All they did was they passed down the nucleic acids, the genes. Our cells then start to manufacture our own proteins. Yeah? So DNA was first, RNA was second, and then proteins were third. This progression of genetic information is known as the central dogma of biology. Again, since biology is able to be changed, we can challenge all the time, and we're always challenging, make sure this is right, do an experiment, challenge the you know the status quo, that kind of stuff. So I think more proper, and the way the book uses it, and this idea of the central dogma of biology is kind of going extinct. We now call it just protein synthesis, the process of forming proteins. So we start again at the DNA level, we convert to RNA, and this conversion from DNA to RNA we're going to call transcription. So DNA to RNA, from double strand to single strand, from deoxyribose to ribose, from thymine to uracil. This change we call transcription. RNA then enables the formation of a protein. So one macromolecule category enables the formation of a second macromolecule category, and that change there is what we call translation. So if you notice, this box is white, this box is white because they are the same family of macromolecules. This box is orange because it's a different macromolecule, a different color, different macromolecule. So the word translation implies two different languages, right? If you need a translator, it's because you don't speak the language. So DNA speaks the language of A's and C's and T's and G's. Protein speaks the language of amino acids. Well, RNA speaks both languages. RNA can communicate with DNA. RNA can also communicate with proteins. So it acts as a translator. It's that in-between molecule between DNA and proteins. So again, this is the preview of tomorrow's lecture. Tomorrow's lecture, I'll get into the idea of protein synthesis. We'll look at transcription, translation. It's a busy process. It'll take a little time to get through that. We'll address then the idea also of genetic mutations and how they have an impact on the evolutionary process for life as well. So with that, I think we made some progress, covered some stuff that should allow you to move forward in your homework now, your exam, and I'll finish this off tomorrow. Yeah. So y'all have a good evening, and I'll be recording some more stuff tomorrow. So y'all have a good evening, and I'll see you next time.